So thank you for having me today. Moving on to something a bit different than what most of you work with, I think. I promised Anders to have a few words on just what the lab does, because he was unfortunately not able to be present himself today. So here at the LGB, we specialize in biogeochemistry and symbiosis, where we combine isotopic analysis with electron microscopy to investigate biological processes at the subcellular level. And we're using this machine here, the NanoSims 50L ion microprobe to do these analysis. And this is what we specialize in, really. And just to give you a brief idea what this is, the NanoSim stands for Nanoscale Secondary Ion Mass Spectrometry. And here we have a schematic overview of the system. So what we do is we label or we spike our biological system with stable isotopes. For instance, we're working with carbon-13 or nitrogen-15. And we cut out small samples of our specimens, and we position them in our system here. We apply a primary ion beam to them. This sputters off a secondary beam of ions that are then collected in this multi-collection chamber. And as the primary ion beam sort of scans across our sample, then we are able to generate these isotopic maps. So we can basically identify which isotopes are present where in our sample visually. And we're doing this first and foremost to investigate host microbe interactions. We're working a lot on metabolic interactions, especially. And at the moment, our lab is focused on cnidarians. We also have a team working on foraminiferous. But in general, previously we've been working with a lot of different species like cuttlefish and nudibranch, and you have mice, plants, basically any kind of biological system that you can label, we can work with in this system. And if you think this is something you can use for your project, if you have something specific in mind, then I suggest that you contact Anders directly and discuss your project with him. So that was just a brief introduction on what we do in the lab. Moving on to an early part of my PhD, so I'm interested in metabolic interactions in the model system Cassiopea, which is a jellyfish. And here I'm going to show you some early results from my, some results from my first part of my PhD, really, where we're investigating the nutrient transfer in the symbiotic jellyfish. So unlike Thomas's model system, the Hydra, that have been around for centuries, the Cassiopea has only been gaining traction as a model system over the last couple of decades, really. So we are really just scratching the surface with the system. The reason that it's gaining more focus now is because that, just like we know from corals and anemones in the ocean, these guys live in a mutualistic symbiosis with the dinoflagellates of the family of Symbiodiniaceae, which is the same family as we find in corals. And this, of course, also means that there is this exchange of nutrients and other services between the host and the symbiont. So for my part of my thesis, I'm interested in, first and foremost, looking at then how much do the jellyfish actually compare to the coral system? Can we actually use it as a model system, as a substitute for corals when we're doing our research? So basically, is this really an analog to the coral as some people are suggesting today? And since this was a new model system for me to work with, I started getting an overview or an idea of how they are actually looking inside, like where do they have their symbionts and all this. And I found early on, working with juvenile animals, so less than 2 centimeters in diameter, that the easiest tissue area to work with from these organisms is looking at the tissue from the bell system. So here we're looking at a transversal section cut out from the small animal. And we have a schematic overview to give you a better idea of how it looks. So it's a very simple structure. We have an epidermal layer that is rather thick, actually. In the middle, we have this vast, the grayish part here, the vast mesoglia, which is what makes up the structure, or like it's the scaffold of the jellyfish, really. So unlike corals, the jellyfish obviously don't have a skeleton. In the middle, we have a gastric network where we have a thin gastrodermal layer, or two layers actually close together. And then we have all these little splotches or dots here. And if we zoom in a bit, this is actually the algae that we have present here just underneath the epidermal layer. And if we go a bit closer now using SEM imaging, and we compare the ultrastructure of the Cassiopea with what we know from corals, we see a couple of key differences here. So again, we have this vast mesoglia in the Cassiopea system, unlike what we know from corals, where they have a relatively thin mesoglia that is usually only a couple of microns. In the jellyfish, it can literally be centimeters thick in adult specimens. Another and more important difference is, of course, that in corals, they primarily keep their symbionts in the gastrodermal layer, so directly in the tissue of the host, whereas for the jellyfish, they actually have them sort of floating around in the mesoglia itself, so they're not in direct contact with neither the epidermis or the gastrodermis. Instead, they kind of reside them in these amoebocytes, which are a specialized host cell that essentially is an amoeba-like cell, which means that they can move around in this mesoglia inside of the jellyfish. And of course, this begs the question now, since the jellyfish doesn't have its symbiont in direct contact with tissue where we would think that it would need the nutrients, does this have an effect on the carbon acquisition for the jellyfish? And this is where the nanosims come into play now for us. So we're doing these pulse labeling experiments where we are artificially spiking the water with a stable form. In this case, I'm working with carbon-13 bicarbonate. So we're replacing the carbon-12 with carbon-13 bicarbonate. And we allow the jellyfish to do, well, they do their thing, and the algae do photosynthesis for, in this case, 2 or 6 hours of incubation time in a controlled environment that looks something like this. And if we dive right into these ratio images that we can get with the nanosim, so now we are comparing the carbon-13 image to the carbon-12 image relative to the natural ratio that we have from a control sample that has not been enriched. If we quantify these images, we get some values that look like this. So now we're focusing on 3 tissue areas or cell types, if you will. So we're comparing the epidermal layer from the host to the dinoflagellates, of course, the symbiont. And then we're looking also at the amoebocyte structure itself, so without the dinoflagellate. And just looking at the ratio images, we can already see from a 2-hour pulse labeling that we have enrichment, especially in the pyronoid structures and in the starch granules of the algae. But we also start seeing small dots appearing inside of the amoebocytes, and these are lipid structures, which is basically photosynthesis that the algae hands over to the amoebocyte as it do photosynthesis. And of course, if we quantify these numbers, then we see that the carbon enters the system through the dinoflagellates. So we have much more carbon overall in the symbiont compared to the amoebocyte and the host tissue. Then if we include the 6-hour pulse labeling, we can see now that the algae is overall much more enriched. So it's been turning over more of its carbon, and it's turned it into carbon-13. The same is the case for the amoebocytes now. It's not just lipid structures that are more enriched overall, but it's also in general the amoebocyte structure that is actually enriched now carbon-13. And we even start to see patches in the epidermal layer as well, from carbon-13. And what both of these pulse labeling periods show us is that, of course, there is a natural transition of the carbon. So it enters the system through the symbiont, which is mostly enriched. It then is passed on to the amoebocytes in the form of these lipid structures that we see highly enriched after 6 hours. And then, of course, the amoebocyte hands over the nutrients to the host tissue itself. But what is more interesting is if we compare the uptake or the turnover of these two incubation periods, What we can see for the symbionts is that after a 3 times longer pulse labeling period, we only have roughly a doubling of the carbon-13 in the system, whereas for the amoeba side, the ratio is almost 4 times as much. And for the epidermal layer, this is almost 7 times higher than 2-hour pulse labeling. And what this tells us, this nonlinear relationship basically, is that coming back to our ultrastructure, that while we do have this gap here between the host tissue and the symbiont, not only do the amoebocytes facilitate a transfer between the two, between the host and the symbiont, but it also does it very efficiently. So it's very good at actually handing over these resources once they become available. Right. So in conclusion, as we saw from the ultrastructure, there are some key differences in the cell or the tissue organization of the Cassiopea, which we have to take into account when we're working with these systems, at least if we use them as a direct model for corals. And of course, as I just showed you with the NanoSims data, the motile amoebocytes efficiently facilitate a nutrient transfer between the host and the symbiont. And of course, this then adds a lot of new questions, because could this potentially have other implications for nutrient transfer in the system when you don't have to use the carbon immediately around where it's acquired, but could it potentially be transferred other places where it's more needed? So really, we're just starting to scratch the surface of potential studies with this new model system. So finally, I would like to thank you for your attention. I would like to thank everybody who's involved in this. This was a joint effort between Anders' lab and the Microenvironmental Ecology Lab at the University of Copenhagen, which is headed by Professor Michael Kuhl. If you want to know more about the study, we have this open access publication out, but you're of course also welcome to ask me any questions. So thank you.
Thank you very much, Niclas. Very nice imaging. Thomas has a question.
Thank you very much, Niclas. That's wonderful. And I'm completely stunned by the mesoglea localization of the symbiont. And I just got so much intrigued because the symbiotic metabolism in this case is, of course, the metabolism of the symbiodinium plus, and this is ignored at the moment in your work, plus the microbiome. And my question would be, do you have any evidence? Where are they? And in the tumor paper which we just got out, we realized that actually the mesoglea and hydra is also a good place for some bacteria to live and to proliferate. So you have, I guess, all the data already in your drawers and in your computer to look carefully, and or we can send you some probes for bacteria, or you have them to look for where are the bacteria in this case.
So this is a very good question, and it is something that I'm interested in looking into. At this moment, we have just, like I said, we're just scratching the surface. There is so much we can do, and I have no clue at all where the bacteria are present, if they're inside the animal or if they're sitting on the surface, like you suggested with your hydra. So if you have something that could potentially help us to identify where they are, I would be very much interested in collaborating on that.
Yeah, just write me a mail.
All right.
Yeah.
I think we have time for one more question from Sebastian Braune.
Hi, Niclas. Nice talk. I have one question. So you compared here the medusum stage of the Cassiopea to the corals, right? Did you check also the polyp stage? I mean, in Cassiopea, as I remember, right, it's relatively special that the symbiont is also in the medusa stage. But I think maybe the polyp comparison would also make sense in respect to the corals.
Yeah. So we did not include the polyps in this study, first and foremost, because we did not have polyps available. But this is definitely something that would be interesting, because the polyp system is much more similar actually to the coral than the medusa itself. But for now, we are only focusing on the medusa here. Because the enlarged mesoglea is a special feature of the medusa in general in jellyfish. So I think it's difficult to compare this to the coral polyp state. I agree. And as you maybe also noticed from my presentation, we don't actually do a direct comparison between corals and the medusa. So it's only anecdotal. You could say that we are sort of suggesting that while it does have this added complexity of the symbionts in the mesoglia, it seems to overcome this issue and work quite well, just like we know from corals.